I'm great to see all your names here. I look forward to chatting with you afterwards. So I'll try to keep it somewhat brief so we can have some time to discuss everything. But I'm not going to so much talk about my personal work, though I'll definitely bring that in as we go along. But I really wanted to just kind of have a check-in with diatoms, environmental DNA, and how I think of DNA and diatoms with the lens of environmental assessment. But before I get to that focal point, the ecological aspect of diatoms and DNA, I want to first give a brief overview of just diatom DNA-based research in general, so that we're all on the same page. I know we've this series has already had some discussion, some guests speaking about some applications of diatom DNA, including Dr. Andrew Alverson's work, which I'll touch on briefly. And I just wanted to make sure everything is clear of what are different methods, what are different applications, kind of for the molecular side of diatoms. Let me share this working. So the brief outline will be that I'll give that brief overview of molecular methods and their applications in diatom research. Then I'll dig deeper into environmental DNA specifically, and I'll define that in just a moment. But generally, DNA that's found in the environment, either in a biofilm or in the water, and I'll extend that eDNA to ecological analysis. And then I'll talk a little bit about some general directions that eDNA is being used in biodiversity monitoring outside of and within diatom research. Um, so getting started on kind of the most common molecular methods and their kind of purposes or applications. You know, the basis of DNA and diatom research really started with using it for taxonomy and evolutionary history. You might be aware of terms such as barcoding or phylogenetics, and Dr. Alverson's talk on phylogenomics. So that's really the core of where molecular methods were first used in diatom work and in biological work in general, more so than ecological assessment, which is kind of the second most common, and becoming increasingly more common, which is the community profiling of diatom assemblages of any assemblage, biodiversity, using such techniques as DNA metabarcoding, quantitative PCR, shotgun metagenomics, and environmental DNA. And I'm just breezing through some of these methods real quick. These are just kind of the buzzwords that you'll hear, and we'll dig into each one of these. And then I'm not going to talk about this third one as much, but there are functional pathways and stress responses that we can get at using molecular methods. And this gets into transcriptomics, differential gene expression, functional genomics, trying to get a sense of what they're doing, how the DNA is actually interacting with diatom physiology and responses to the environment at that cellular genetic level. I'm really not going to speak on that today. I'm going to focus a little bit on that history of its use for taxonomy and evolutionary history, and then dig deeper into the community profiling side. So, so DNA barcoding is a word that you'll hear a lot when you venture into the DNA world, and you'll hear barcoding and metabarcoding. And so I want to set the record straight on, barcoding itself. And DNA barcoding is really focused on single species, a single isolate. And when we're talking about diatoms, we're thinking of a single cell culture or a single cell pick. And then we extract the DNA from that cell or that culture. Then we focus on, I hope you can see my cursor, but basically you'll have a double-stranded DNA, split it apart, and then we attach what are called primers, which are conserved regions of a DNA sequence, so A, C, T, and G, but it's unique to this gene and to this target target taxon or group of taxa that you're interested in, but it's conserved. And then there's enough variation in between. Sorry. in between that primer, those primers, so you have a forward and a reverse primer, and that region in between is what we're interested in. That's going to be the barcode. That's basically the unique sequence for that gene of that species. And we amplify that gene region, we sequence it, and then we process those sequence and validate that species identity. So we add in a primer, which will then synthesize, allow the synthesis of the actual target gene region that we're interested in, which allows us to get at species level identification or even intraspecific variation, genetic variation, whatever we're interested in. That's what a DNA barcode is. And when we think about using these for evolutionary history or phylogeny, these barcodes are usually anywhere between 500 and 1,000 base pairs. So 500 to 1,000 A, Cs, Ts, Gs. And in diatoms, we're usually looking at a couple of different gene regions like RBC, that are often in the chloroplast, like RBCL or ITS, or some nuclear ribosomal genes like 18S. When we find, and it's been shown that those gene regions are kind of phylogenetically or evolutionarily informative. They tell us something about the species ID and the variation across taxa. And where this is used is we can use it to, one, validate species identity like that previous slide showed. Also resolving cryptic species, detecting novel lineages, tracking that evolutionary history, that genetic structure across space. If we're looking at a single species, how different populations of that species vary over space and time. And it supports our ability to use it in other applications for multiple species by contributing to reference databases. This basically becomes our accessioned specimen of genetic specimen of a given species at a given location. And we can now use that information to, when we have an unknown sequence, to relate that back to a known taxonomy. So, like I said, there are a couple of different markers that are commonly used for DNA barcoding and understanding species genetic variation within and across species. And like I said, this UPA here at the bottom is also 23, called 23S. That's a chloroplast gene. Then you have a ribosomal gene, RBCL, and then cytochrome oxidase, CO1. And you can see here the black bars show whether it can be detected, and if it extends across different species, shows that it can't differentiate across those species. So for instance, this UPA can't differentiate Nitzschia brevirostris from Pusilla and laevis, whereas and none of these really can. In CO1, the empty white bar shows that it doesn't detect it at all. That primer region is likely not amplifying it because of variation in that primer region itself. Whereas UPA and LSU are able to detect and differentiate Nitzschia alba from other Nitzschias. So right away we have a decision to make when we do DNA barcoding of which gene are we going to use. So they're decisions that we have to make. So this all has come together in a great resource. It's a curated reference database called diatom that was started in about 2012 from a group in Europe as part of called the DNA AquaNet initiative, which was to integrate molecular methods into the Water Framework Directive national or international monitoring programs. And that reference database has ballooned from about 1,000 barcodes from the 2012, 2015 time to about 9,000 barcodes from over 1,700 diatom species, largely concentrated in Europe, but also across the US and some in China and Japan. But you can see the scarceness of those barcodes from other regions and the need to develop that more potentially. So where we can apply DNA barcoding, you all may have heard of the term of phylogenetics, and this is really extending barcoding to multiple genes and multiple species. So now we can align barcodes from different taxa, and here, just a quick rundown of, butterflies versus owls, and each of those colors, green, red, black, and blue represent the different nucleotides present in DNA of A, C's, T's, and G's. You can see there's some variation there. So what we mean by aligning is to just line it all up, position one of each butterfly of each owl. We line those up and then we look at dissimilarity of that alignment essentially. And that's what a phylogeny is. And we can visualize that in a phylogenetic tree, and we can support that tree, how confident we are in the different breaks in that tree of how different species are genetically from those barcodes, and then create this dendrogram and compare clades both genetically and then validate this with morphology as well. How a real world example of this in diatom research is a great study by Evelyn Pencil back in 2019, where she and colleagues looked at Pinularia borealis populations from across the world. You can see some in Antarctica, a lot in Europe and Greenland, places like that, and looked at morphometrics and ultrastructure of Pinularia borealis and I think three or four barcodes, so genetic barcodes, and and found that there were eight distinct genetic lineages, but that those lineages were not reflected by the morphometry. Basically, they were indistinguishable morphometrically, ultrastructurally, but genetically they were quite distinct to the point that at least one of them was close to being described, if not described as a new species. So it shows the power of DNA barcoding, and looking at just a handful of genes to be able to see differences where more, classic approaches like morphology may not be able to find differences. And it's an open question that we all need to consider and often struggle with if you're in the DNA world of, to what end do we integrate this genetic information into our concepts of what is a species, and what does variation, across space actually look like? And are these meaningful differences or not? These are questions that the field still wrestles with a bit, though increasingly we're finding robust support for the molecular side, and the differentiation of barcodes is supportive of some ecological difference as well, and some species level differences as well. And then we can extend this beyond genes. So barcoding is focusing on small regions. There are hundreds of millions of base pairs in a diatom genome. And we're only looking at 500 to 1,000, maybe 1,500 in this example to differentiate these different lineages. Is that sufficient? So there's an approach of phylogenomics, which you might hear shotgun genomics or genome skimming, to refer to the total DNA, of sequencing the total DNA from a culture, or transcriptomics, which is looking at the mRNA, basically the expressed genes, to get a sense of larger scale changes across the genome. And this is not just these phylogenetically informative markers, these classical ones like the UPA or the LSU or CO1 or RBCL, but functional genes as well. So it allows you to dig a little deeper into different aspects of the diatom genome. Um, and basically, the process for running shotgun genomics is that from a single species, you fragment all the nucleic acids into many short pieces. You sequence everything. There are no primers. You just get everything back. And oftentimes, you'll get some bacteria, things like that. But then you'll assemble those sequences based off of their overlap with each other, and oftentimes with some reference that we have available of a closely related taxon. And then we can actually assemble a full genome sequence and then annotate that based on which genes we retrieved from that process. And this was really this process of genome skimming is a shallow whole genome sequencing, essentially, where we're not, in order to do this process of assembling a genome sequence, you need a lot of sequences. And to do a whole genome that way requires a lot of sequencing depth that oftentimes is too much for most studies and might be overkill for certain applications. So genome skimming is the process of doing this fairly shallowly and getting the overrepresented genomes of chloroplast and mitochondria, which also allows you to get the barcodes, which is a really a great way of double dipping, if you will. So you can get a whole chloroplast genome, 150,000 base pairs in this case of Halimphora, and you get all the different genes that make up that chloroplast genome, including those classic barcodes. So you can understand old school phylogenetics and plug and play into those, but you also get a really powerful data set that if you compare chloroplast genomes, you can start thinking about things like speciation, and that evolutionary history a little bit. And this was done in a really elegant way. And this is what Dr. Alverson and colleagues talked about earlier in this Web Academy of using transcriptomes from thousands of genes from hundreds of diatoms to basically reconstruct evolutionary transitions of diatom lineages, proposing almost a new phylogeny and evolutionary history with a timeline integrated with fossil records of diatom divergence, essentially. So I highly recommend that talk and that paper. It's a really fascinating approach, but this is getting into the core of where did diatoms come from? How long ago did these speciation events occur? And they're doing that not with a couple of 500 base pair genes, but with 1,000 plus base pair genes from thousands of genes from hundreds of taxa. So a very powerful data set that they were able to compile there. And that's one of the great powers of molecular approaches with diatoms in general is that, it's a constantly building data set. Any new project is contributing. Usually these data are made publicly available in such resources as NCBI GenBank. And even if your goal isn't to do transcriptome analysis to reconstruct evolutionary transitions, you can still pull down functional genes from these projects or classic phylogenetically informative genes for your smaller little study, things like that. Um, so I'm just making sure. Yep. Yeah, and I see that Sarah put that seminar in chat, so please check that out. So this is the classics of diatom molecular research. But it's really in the last 10, 15 years taken off in a new way, which is more community profiling and ecological assessment. And this is where we're adding meta to everything. This is the new meta to be hip and cool. But DNA metabarcoding, shotgun metagenomics. And when we say meta, when I say meta, it really just refers to everything, everything in an environmentalist sample, essentially. And so instead of a single culture, a single cell pick, it's a biofilm, and we're sequencing everything in that biofilm. And that's where those reference databases come into play, is because and this hard work that was done by all of the phylogeneticists and the like, the DNA barcoders, that they really laid the foundation for DNA metabarcoding and metagenomics to proliferate, because now we know what these things are. We're not just going in blind, essentially. I'll also touch on quantitative PCR, which is more of a single species targeted assessment. And then this all kind of the umbrella for all of this is eDNA, or environmental DNA. So collecting DNA from the environment. So DNA metabarcoding, again, collection of barcodes, essentially. So we're, instead of extracting DNA from a culture, we're extracting it from an environmental sample, whether it's a biofilm or a water filter, for instance. Then we barcode essentially all target taxa. And I mentioned the primer region before needing to latch on to that gene that we're interested in. Now we have to make sure that primer region isn't just specific for the species, but is specific across all of our target taxa. So if we're interested in all diatoms, we need to make sure that that primer region. So here, this is a little snippet. This is actually for arthropods, but this 20-ish base primer region, the C here is in blue. It's saying all the non-target OTU, all the non-target taxa in this study also had a C in that position, but our target ones did. But if we jump to the very end of this primer region, you can see there's this one base where all of our targets have this C, which is part of this primer sequence. Most of the non-targets don't have a C there. So it's not going to amplify the non-targets. It's just going to amplify our diatoms or our insects or whatever you're really focused on. And this way, we're not amplifying a bunch of bacteria. We're only amplifying the diatoms or whatever your target is. Generally, these barcodes are shorter than for phylogenetics. Phylogenetics, classic DNA barcoding was done using a technology called Sanger sequencing predominantly. And that allowed you to sequence a bit longer of a barcode, 500 to 1,000 bases. But metabarcoding is really applied using next generation sequencing or high throughput sequencing. You'll hear of Illumina, for instance, or nanopore. And Illumina specifically has been the dominant sequencing technology for the last 15 years for metabarcoding. And it can only accommodate no more than about 450 bases. So now we have an added challenge of shortening that barcode and still having it be informative. But oftentimes for metabarcoding, the goal is not to reconstruct phylogenetic trees, it's to identify taxa. So do we need to know everything about that taxon to be able to differentiate it from other taxa? And that's where those reference databases come in. And being able to assign taxonomy based on something we've already seen before using barcoding or genome skimming, things like that. Or there are applications of DNA metabarcoding where you don't, apply taxonomy at all, and you're just focused on these unique sequences, and you do a taxonomy-free approach. And those unique sequences are often called ESVs or ASVs, amplicon sequence variants. But you can think of them as like kind of denoised or error-corrected unique sequences. And a final note on DNA metabarcoding is that it generates compositional data only, so it's relative abundance only, at least historically. And I'll try to touch on a little bit of where that is changing. And this just shows you that breakdown of instead of just a single taxon here, we're breaking apart all of those, all of the all of the DNA in a sample, and we're trying the DNA barcoding is just looking at one taxon, metabarcoding is everything. Here's just the general process of walking through with collecting. This is from invertebrates, but imagine a rock scrape, and then you sequence that, or you take a filter water, take a water sample, and you can get that. You get a barcode database. You basically get a count table of taxa and relative abundance, and per sample. And then you can do it the old-fashioned way with morphology. And I say old-fashioned in a loving way and not in an antiquated way. It's still morphology, as I'll touch on, is a critical part of this whole process. Just like barcoding laid the foundation for metabarcoding, morphology laid the foundation and continues to push forward all of this diatom research. I'm not going to touch too deeply on shotgun metagenomics. Just know that you can basically, do this without primers and just amplify everything. But this gets tricky with diatoms because in a mixed environmental sample, oftentimes it's not diatom dominated. You might get a lot more bacteria, or if you're in a phytoplankton dominated system with a lot of cyanobacteria, you might get more cyanobacteria back. So you can get kind of swamped out of your target using shotgun metagenomics, but you have the power of not just being constrained to that one little gene region. You get all sorts of different genes, including functional pathways, and that has its own power. So an example of shotgun metagenomics is some work that looked at seafloor sediment diatoms and found comparing shotgun. They winnowed the field to only this RBCL region using shotgun data and using amplicon or metabarcoding. Amplicon is that amplified region from metabarcoding. They found pretty distinct community composition between shotgun and amplicon and pretty distinct taxa as well, not a huge amount of overlap. But that said, shotgun data did detect more rare taxa, whereas amplicon data kind of ran away with really abundant stuff. The abundant stuff dominated and drowned out the non-abundant diatoms. Okay. And then finally, I'll just touch very briefly on quantitative PCR. It's really not used too much in diatom work. It's going back to species-specific environmental detection, essentially. But basically, you can amplify target DNA in real time by attaching a fluorescent probe, essentially, to the DNA strand. And you measure that fluorescence as DNA is synthesized. And that translates to a quantified DNA copy number, so absolute abundance of DNA copies. And this allows you to quantify a specific taxon and maybe even detect or focus on low abundant species that might be swamped out, like in that previous example, and allows you to get that absolute abundance. And it's not used too often, but a great example in diatom work is looking at Cymbella genishii, an invasive species in this location here in China, in Chinese rivers, and found that microscopy, benthic qPCR, so rock scrapes, and water eDNA, all correlated very strongly with detections of this Cymbella. Basically, where microscopy found it, you also found it using qPCR. So it's a great, but you also added, maybe in some cases, a little extra sensitivity than microscopy. But it's a why use qPCR instead of microscopy would be the turnaround time. It can take only a week or two to do the qPCR here, and cost, maybe $50 a sample at a commercial rate. So, moving on to some specific applications of environmental DNA and DNA metabarcoding in an ecological context for diatoms, which is really where a lot of the emphasis is right now. A lot of the energy in diatom molecular research is. I'll point you to these two papers. David Williams and Lauren Ball's are, comments, essentially mini review and a comment, talking about, does it matter what Fragilaria is called, metabarcoding in the science of taxonomy, where David Williams kind of argues for, classic approaches and exercising caution with the application of barcoding and metabarcoding in how we think about identifying diatoms. And that was contrasted with a response from Lauren Balls of an ecologist, like an ecological perspective, putting metabarcoding as a diatom identification tool in the context of where it's most applied, which is in environmental assessments, general ecological patterns and responses to stressors, environmental pressures. And in that case, arguing that it's more than sufficient to get at what you want. So it's when we think about DNA for diatom research, it always comes back to what is your question and is it sufficient to answer that question? And I always advise everybody to think about that. What is the right tool for the job, essentially? Because at the end of the day, in the DNA metabarcoding context, it is a tool. It is another tool in the toolbox, and we can talk about whether it's necessary to spend so much time developing a new tool. But we're finding that there are applications that morphology is not able to do, at scale or at cost, or with the consistency that metabarcoding can. So it begs the question of, is there utility in this newer technology? And we're seeing across environmental or ecosystem types, across organismal groups, that environmental DNA studies are increasing exponentially over time, to the point of, having a couple papers, in a couple dozen papers in 2008 to over 500 a year over the last couple of years. I mean, I get alerts every day for 10 new metabarcoding papers of, whichever group. And whether that's a fad, great question, But let's think about whether it is useful for diatom research. So I'll just make some broad claims right off the bat, and then I'll add some nuance to it here at the end. But diatom metabarcoding has been shown, especially over the last 5 years, to be an established, robust method for a lot of applications. So we can see this is just diatom publications and citations here on the right over time, and that it is following the general trend of environmental DNA and DNA metabarcoding more generally. But I'll speak a little bit that methods are reproducible across labs, which was a big question mark even just 10 years ago. Um, it supports general ecological trends, those broad scale patterns. In general, it's consistent with morphology, those broad scale patterns, but maybe not the specifics. So there's a bit of an asterisk there. And at the end of the day, it's already been integrated into water quality monitoring programs. So it's being used. To me, that's a great validation of it as a method in and of itself. So thinking about the reproducibility across labs, there was a ring test that was held across 17 labs, and the top two are using a fixed method. The top two rows here, E1 and E2, and you can see across labs, everybody was following the exact same extraction and PCR protocol. Extraction top, second is PCR protocol, and outside of one lab that had an error. There's a very similar community composition of diatoms from river and lake samples. And then when labs were allowed to use their own DNA extraction method, you still see incredible similarity across those labs. And same thing for their own PCR processing. So composition is robust to all. There are so many decisions that have to be made in the DNA processing workflow, and I'll touch on them a little bit. I've previously talked about this oh my goodness, about 5 years ago. There was a Diatom Web Academy about the actual lab processes and where we were. So I reference that if you want to dig deeper into the actual methods that underlie these final results. And then thinking about general patterns, I'm just throwing a couple of things out there. I'm sure you can find other examples where these don't come out so strongly, but that they are coming out is some validation of this method. But in this study, they looked at DNA metabarcoding across assemblages of different climatic zones and found, at the very least, you're seeing differences between tropical, temperate, subpolar, and across tropical islands even differentiate largely. So expected ecological trends are reinforced through this metabarcoding approach. I saw Eric Pilgrim's on the line, so he had a role in this work with Nate Smucker and colleagues at the EPA. They've taken biofilm collections as part of their um, national aquatic resource surveys over the past couple of years of sampling, and then processing those data so for meta barcoding. And some early results are showing pretty strong relationships between some classic local environmental predictors of diatom composition like conductivity, total phosphorus, total nitrogen, pH, that when we look at an NMDS axis, an ordination axis of community composition, that there's a strong relationship between these environmental pressures and community change across ecoregions across the US. I did at Jonah Ventures, my colleagues and I did something similar using a different genetic marker or a different barcode, and using environmental, water, aquatic environmental DNA from a water filter and didn't look at those local stress pressures, but looking at landscape variables. And both of these oftentimes play into aquatic assessments, and found that, space or geography was the predominant driver of diatom community composition and other algal groups as well. But also an index of catchment integrity, basically darker shades here in this heat map show a stronger effect on community compositional change. So that diatom metabarcoding derived assemblages were affected by things like stream order, catchment integrity, and space across the scale of the US. Okay, all that said, there are, I'll get into a little bit of the nuance and then a little bit of the application to environmental assessments specifically and why we're interested in using DNA metabarcoding. There's always a question of how does this relate to previous data sets, to previous methods. We want to make sure that as I've shown in the past slides, that there is good concordance, but in community composition between metabarcoding and expected patterns. But what does that actually look like on a one-to-one comparison with morphospecies, morphology-based assessments? And we can see in this one study of comparing microscopy to two different genetic markers that species richness was much greater using this RBCL metabarcode versus both the 18S and microscopy values. Yet, when you look at the actual identity, you see that there's not a huge amount of overlap of what those taxa actually are. Microscopy, 18S, RBCL were basically distinct taxa. And when you look at that in an ordination space, that falls along the line where the method is driving the relationships among sites, not the actual site. We're not seeing clustering of sites here. We're seeing clustering of method. And that is a big question because previous studies have shown that even with morphology, that we have where taxonomic or taxonomist bias or taxonomic inconsistency can be the dominant driver of compositional differences more so than the actual environmental pressure. So who is identifying the diatom has a larger impact than some of the environmental drivers that are expected to be the more important variables. Yet, that said, despite those differences of, not a complete alignment one-to-one of microscopy and metabarcoding or even 18S to RBCL, we see when we apply it to an index of biological integrity, which is a tool for environmental assessment, basically whether a stream or a lake is healthy or not. In the bottom left quadrant, we're seeing, healthy conditions or a healthy environment in the upper right, an impacted or a disturbed environment. And on the x-axis here is the morphology-based index, and the y-axis is the DNA-derived index using assigned taxonomy on the left column, taxonomy-free, so only using ASV as a taxon unit, or using a fancy machine learning protocol. And you see generally a very strong alignment in the actual ecological assessment between morphology and any of these metabarcoding, any of those metabarcoding genes or applications. And this kind of plays into where we are in environmental biomonitoring in general. So this is the morphology-based work. And I think Luisa, I saw you were here as well. So I'm gonna please chime in and chat if I misrepresent this at all. But basically, there's been some work shown across the US using these multimetric indices of diatom traits and compositional metrics, if you will. So trying to make sense of this hyper diverse diatom community to be able to use it for environmental assessment of whether they're changing from disturbed to reference streams. So in white here on the left are the least disturbed streams, and the gray are the most disturbed streams. And this work showed here on the left um, these were genus-based multimetric indices that only genus level taxonomy was needed to differentiate these least disturbed and most disturbed sites, and we can apply this model that's developed from only genus level traits and still get accurate delineations of ecosystem health. And on the right side, we're seeing that they showed that fewer counts, counts of 100 valves instead of 600 valves, were just as adequate for differentiating those least and most disturbed sites, and could be applied across these regional indices to differentiate or to assign stream condition. So less effort, which might play into which helps for morphology-based work for sure, but also might allow for increased utility of DNA metabarcoding, where taxonomic resolution might be limited because of reference databases. This might allow more plug and play of metabarcoding into existing tools. And oh, let me check something. Yeah, and this is some work that I participated in with Sarah Spalding, with Darren Carlisle, and others from the USGS and EPA, that showed that large-scale diatom MMIs still need to be regionally constrained. Looking at, you can't do a whole large-scale conterminous US single index. We needed to create different indices for the West, for the plains, and for the Eastern lowlands. And this goes back to that work that I showed earlier that we see that DNA metabarcoding shows that for diatoms, that space is a large driver, so that we're seeing that consistency in metabarcoding results to what we're seeing with morphology. That space is still a big driver, so that we can leverage what's already been done in the morphology space to integrate this stuff in with metabarcoding. Yet I want to be clear that metabarcoding still comes with nuance, as I just said about morphology versus microscopy. There are also things about those decisions as throughout the metabarcoding workflow. Recent papers, as recent as just a couple of weeks ago, that are coming out about the effects of collection and storage duration on composition, on extraction method, on which gene marker you're using, on how many PCR replicates, how much sequencing depth, which bioinformatics pipelines you're using. Those all have an effect on on that final community composition, on which species are detected. But over and over, just like with that ring test I showed, those different methods are still leading to similar ecological conclusions. And that is what is the difference. If you're looking for complete truth with metabarcoding, you're not going to find it, in my opinion, because there are so many different decision points. Different methods are going to lead to slightly different results. But in general, these broad scale ecological patterns, and even some very nuanced patterns, are able to be retrieved with metabarcoding. I talked about the incongruencies with morphology already, and I've touched on a little bit that the reference databases from that early barcoding work are still inadequate. This is all algal freshwater morphospecies that are used for environmental assessment. But you can see that, of the 5,200, only 200 of them were in GenBank a couple of years ago for this one gene region. And then finally, standardization is lacking, and that can spook a lot of users. I'm running short on time, but I want to just quickly mention in the last 2 minutes I have here before we open it up for discussion, some cool directions. And I won't be able to dig as deep into this, but hopefully it'll whet your appetite a little bit as to the neat things that I'm able to do, where I'm working at Jonah Ventures in this DNA service lab in support of our clients, but also just for DNA research in general, and just the directions that biodiversity monitoring is going using environmental DNA. So quick, I'll talk briefly about sequencing technologies, making metabarcoding more quantitative, doing some cool target enrichment stuff, and then looking at airborne DNA. So you heard me mention Illumina as the dominant sequencing platform. There's a relatively new sequencing platform called Nanopore, which is really built for its ability to do longer reads than those 200 to 400 that Illumina is so good at. But we found that just looking at those short reads, one of the big things with Nanopore is that it had a higher error rate. But over time, that error rate has gone down. So we're getting more accurate sequences off of the sequencer. It's a lot cheaper to use, a lot more user friendly. And we're getting relative read abundance between Illumina and Nanopore of an incredibly high R squared of 0.98. And really the differences between Illumina and Nanopore are really low abundance stuff. So by and large, what we've done at Jonah Ventures is actually moved over to Nanopore sequencing instead of Illumina because of its ease, because of its ability to go a bit longer read as well, while without sacrificing species detections and sensitivity. I won't get just kind of ignore this detailed diagram here on the left a little bit, but there's um, another way of what we've been trying to do is make DNA metabarcoding more quantitative, so it's not just relative abundance. And basically, we add a spike in of a known quantity of known DNA, and then we use that as a scaling factor after sequencing to get back what the actual DNA copies of everything in the sample was. We did this with some goldfish, looking at goldfish qPCR copies versus next generation sequencing copies. Again, very strong R squared between those. So we're seeing that we're able to move from compositional to quantitative with metabarcoding. Another quick thing, I won't go into the details either, but basically we can imagine throwing in a bunch of DNA seeds for all the taxa that you're interested in. And instead of trying to find a single primer that amplifies all diatoms, you're like, I have 10 diatoms I need to find in this sample. And I want to do it across the whole chloroplast genome, and we have those genomes. You can get these little probes, these biotinylated bait probes, and enrich the target DNA and just bind to those probes and only get the target taxa that you're interested in. A study did this with mosses and found that it has kind of mixed results, but the idea is that you enrich in the DNA that you really care about. It's basically an alternative to shotgun sequencing where you're getting a bunch of off-target stuff. It allows you to get all on target, but a variety of different genes. And then finally, airborne DNA is a pretty crazy thing that offers a way of moving environmental DNA into the terrestrial systems. A couple of cool papers. One detected 49 vertebrate species at the Copenhagen Zoo with just a couple of air filters. So we're seeing that things are, dispersing DNA off of them all the time. Some work Jonah Ventures did is go to Panama, Barro Colorado Island, and found just with a couple of basically computer fans and with some MERV-rated filter paper, finding thousands of insect species and hundreds of vertebrate species. And this is even relevant for diatoms and thinking about dispersal. I had a paper where I worked on Antarctic diatoms and found passive dispersal of diatom RNA and DNA across the McMurdo Dry valleys, and that it was a signal of local dispersal. So we can get at some of these community assembly mechanisms in addition to just compositional changes. So a quick summary that molecular methods can be used for a variety of reasons. DNA metabarcoding is a robust method for yielding expected ecological patterns and assessments, and that biodiversity monitoring is increasingly integrating environmental DNA. And with that, I apologize for not having a lot of time for questions, but I'll take any that we do have time for.
Thank you, Nick, for a wild ride across all sorts of terrain with DNA.
Yeah.
Um, and we've just got a few minutes if anyone would like to raise their real or virtual hand with a question for Nick. Yeah, please go ahead. David's got a real hand.
Real hands. Hey Nick, thanks a lot. That was a really great overview. I have one quick question for you and then another. Where can we find you and your diatom songs on Spotify?
I think it's only released on CD. I don't remember who has that CD though.
I think that lives in Montana. Okay, so you touched on this a lot, but as a lot of us are not, well, shall I say, geneticists, what are maybe some common pitfalls you see or areas you would exercise caution and not like overinterpreting metagenome data or
Yeah.
metabarcoding, rather? Sorry.
Yeah, I think it's a great question of, and I touched on it briefly with those nuances and, trying to recognize those limitations, that's compositional data, that there are, as you dig deeper, there are, effects of sequencing depth. If you don't have adequate sequencing depth, you might be missing taxa. So you can't take it as, complete species richness, exhaustive species richness. Same thing with PCR replication, so understanding the limitations of the approach you used, how many replicates you had, how much water volume you filtered, things like that. Um, just knowing that those could lead to limitations, and maybe they don't. But that's where having a kind of a pilot study is always great if you have the time for it, because you get a sense of what some of those limitations might be, and then validating it against ground truthing it against some classic methods like morphology. Yeah, Mark.
There you go. Got myself unmuted. Thanks, Nick. I appreciate it. I gotta jump on to another meeting here, so I'm only going to be on for a couple more seconds. But one thing I wanted to ask you was, what kind of advice would you give young up and coming people in terms of what they should be looking for as this field is progressing so rapidly? What should they be training themselves for? What should they be ready for?
Hmm, that's a great question. I think I would recommend still, staying grounded in, the question, um, and don't get too deep into the methods of it per se, right? If, use it as a tool, understand how it's used, those limitations that, David asked for, what it can do, building up some of those some confidence in using those methods, understanding the process of DNA extraction, of PCR, of sequencing, those are more and more accessible even in the classroom. So taking advantage of those opportunities, but not losing sight of why you're doing this in the first place. It's not a silver bullet. It's not a catch-all. There has to be a purpose behind using this. And if you're just doing it, I mean, right now we're still in the phase where you can get an easy paper with just saying that you've done DNA but metabarcoding in a new environment. If that's your goal, then do it, but do it, with intention, and recognize that you stay grounded in microscopy, in diatom autocology. So understand how this actually reflects the real world, if you will.
Okay, thanks.
Yeah.
Yeah, Layla has a question. Would you like to come on
Yes,
screen and ask?
hello. Thanks, Nick. I missed at the beginning. I'm sorry. I wanted to ask you if you can use this method to find the Jurassic or Cretaceous diatom. Thomas and Pat said that the first diatom are probably in 274 million years before. So while we are trying to find the oldest diatom and to understand, is it this method? Is it possible because we have some Jurassic with just clay and some? I mean, there are some samples that we might try to see if we can find the oldest diatom. What do you think? Thank you.
Yeah, it's a great question, and I'm not as up to speed on ancient DNA of diatoms of how far back we've been able to get DNA, actual DNA from. But I know, in other organisms. It goes back at least tens of thousands of years. I'm not sure. I really, it's not my area, but I know, like Dr. Alverson's work showed, that you compare it with the fossil record and look at phylogenetic change and infer those differences. But actually getting that far back, I'm not sure how well DNA is going to be preserved in the environment. We have, gone back tens of thousands of years, I think, for diatoms alone, but I'm not sure about, that scale, that timeline.
Okay, Thomas, you don't have an idea because you about your paper that where you changed all the classification? No, sorry. Thank you.
Yeah.
Oh, are you asking Tom
Yeah,
that question?
yes, yes. About the change of their classification, did they use the same method or no? Oh, I'm sorry.
Yeah, that was the like, pairing fossil record diatoms, in taxonomy with, genetic divergence, I believe. But it wasn't, as far as I'm aware, it wasn't actually pulling DNA sequences or RNA sequences from, ancient individuals. And that,
Okay,
that's its own challenge. Oftentimes DNA is more degraded too. So you would, get very small fragments that might not be informative. So that's where, the power of microscopy, traditional methods, paleolimnology will always have its place for those sorts of questions, I think.
thank you very much.
Okay, I see that Sarah left. Sarah from Evelyn Geiser's lab, but her question is in the chat. And
Yeah.
so let's just put it up for her and she can go to the recording. On the slide where you compared the different methods, and I think that she's referring to the Illumina versus nanopore,
Oh,
you
mhm.
only compared the two types of metabarcoding. Are you a prominent, are you a proponent of getting rid of microscopy as a method of identification?
Yeah, no, I'm not a proponent of getting rid of microscopy. I think it serves, I mean, as we were just saying, very, in paleolimnology especially, serves a great purpose. But even, there's a massive record of morphology being used for environmental assessment for long-term data. And at the very least, we need to make sure there's a transition of understanding those differences between morphology and metabarcoding before any complete shift away from morphology would happen. But I don't think I mean, I don't see on the horizon a time when morphology will be defunct as a method. Maybe for certain programs where there are limitations of, cost and scale, perhaps, but there's still always going to be a need for understanding, what these things actually are. And genetics can only go so far for that. And why I would only compare Illumina versus nanopore is we just don't do diatom taxonomy at our DNA service lab. So we didn't have any ground truthing, and that wasn't diatoms only. But that work is being done. Most studies before implementing DNA into ecological studies do that comparison at the minimum, and almost every paper shows them to be complementary methods, not replacement methods.
Yeah, Tom, go ahead.
Okay, yeah, yeah, great education, Nick. I appreciate it. You showed that graph. We showed the eDNA studies of increasing exponentially over the past few years. But it doesn't seem like the metabarcoding library, at least here in the States, has kept up. So I mean, we seem to need an initiative like they have over in Europe to, build that library up. And you have any suggestions for how we can promote that and get that done?
Yeah, I'm not so much. I think, it's a similar issue that we find even with, cataloging the morphological diversity of diatoms with DONA, right? Of, trying to invest in this foundational knowledge. It's harder and harder to find those funders and those individuals who um, can spend their time doing that. And one thing I've thought a lot about is, with the cost decreasing of things like genome skimming or just, nanopore sequencing, that these are more accessible methods for smaller labs that might not be grant dependent, or grad student projects that really just take time and relatively few resources that can be provided at, within university grants. If a grad student worked on a species page, but also, coordinated with culturing that taxon and sequencing it, can be done at a pretty small fee, or small cost for them doing it themselves or sending it off. So I could see the kind of the democratization of it rather than expecting some large scale, funding support. But, the Smithsonian Institute is really it emphasizes a lot of biodiversity, like kind of dark taxa identification. They're really emphasizing biodiversity surveys using DNA for fishes and marine invertebrates. But trying to get them into more of the diatom space, I could see a resource like the Smithsonian, maybe, getting pitched that, or, those who are pushing the large scale monitoring surveys, states, federal government, that there is a need for that taxonomy based those taxonomy based indices, though that, as I showed, that has its own question mark. Do you need good taxonomy for those applications? And therefore, those users might not support building out those reference databases because it's not pivotal for their questions. But I think democratizing it is one great way, just like we do for like DONA species pages. Oh,
Thank you, Nick.
You're on.
yeah.
Yeah. Thank you, Nick. Maybe someday diatoms ought to work, and give some incentive to get the sequence at the same time as a page is done. So, okay.
Yeah, and it would be
Thank
great
you.
to do some more like single cell cultures and, their way, flow cytometry where you're kicking out, individual cells into a PCR tube. You do single cell genomics and you get it right off the bat. That is possible. And, just takes somebody with enough know-how and interest to do it.
One last question, quick question. How much does it cost per sample for eDNA? Let's say if we
That
wanted
was
to propose
my
it.
question.
Oh.
Yeah.
Yeah.
So just to give you a quick snapshot at like a commercial rate at Jonah, we charge roughly $100 a sample for metabarcoding for a single primer set. And then if you go to multiple primer sets, it's, a $25 discount for each one of those. qPCR is about $50. DNA extraction is about $20 at this commercial scale. But, if you were doing it all yourself in a lab, you could get away with doing it, a bit cheaper. But the cost savings really does come in when you're doing 100 samples, for instance, right? If you're trying to run 2 samples, it's going to cost, in-house, it might cost you more than that even. So scale is your friend in that case.
Thank you, Nick.
Yeah.
Well, we're past the top of the hour, so thank you, Nick, for your presentation today, and thank you for everyone for joining for the final episode of Diatom Web Academy for 2025. Wow, look at all those applauses going off. Take care, everyone. Have a great holiday, and we'll see you in 2026.
Thanks, everybody.